Now we will talk about synaptic transmission, how a neuron communicates with another neuron and the effects of drugs on this process. So a lot of this is going to be a review of what we just talked about when we talked about the anatomy of a neuron, because I kind of told you the different parts and how they worked. So first, the axons branch out and end near dendrites of neighboring cells. So if you remember the anatomy of a neuron, you have the branches, the dendrites receive the information, the terminal branches and buttons, they send the neurotransmitter the information to the next neuron. So axon terminals, right, are the tips of the axon branches, right? So those are the terminal buttons. There's a gap that separates, the axon terminal from the dendrites. So even though it's extremely tiny, there's a little gap there. That's called the synapse or the synaptic gap. Usually you see the synapse, but that's that little space in between the two neurons. So the steps to synaptic transmission. Action potential causes synaptic vesicles to open up. And I'll show you a picture of that on the next slide. A neurotransmitter, NT, so from now on you'll just see NT, know that that's a neurotransmitter, is then released into the synapse. The neurotransmitter fuses to the receptor site in on one of the receiving dendrites. The receptor site then opens up and allowing positive sodium ions to enter the dendrite, triggering action potential in the next neuron. All right, so these neurotransmitter, it's kind of like a key. And I'll show you a visual in a second, where it opens up the dendrite, and that's when that threshold is passed, if you remember us talking about that before. So the threshold is now passed, where the neuron, the connecting neuron, where it was polarized, now it becomes depolarized. You have all those positive sodium ions rushing in, and that's when you have action potential. So now that neuron has just fired, and then that's what it keeps doing, one neuron to the next neuron. So here's a visual, right? So you have the sending neuron coming down here. Action potential has happened, so it's firing. If you remember those gates opening up and closing. If you remember the neuron can't fire again until it recharges itself. Those gates have all, repolarized. That's called refractory period. So you have these vesicles containing neurotransmitters. So these sacs, right? These are called vesicles, and the neurotransmitter is the actual, message that's being sent. They're inside all these different sacs. All right, they're inside all these different sacs. So they get down to the terminal branches, down to the terminal button. This would be the terminal button here. And this little gap here is called the synaptic gap or just the synapse. And then it releases these neurotransmitters. These are these molecules, right, that say, send a message that wants to be sent. If you're in pain, maybe it's a pain relief thing. And then it connects to these dendrites. So this right here would be the dendrite receiving these neurotransmitters, and they go in here and they open up a gate, right? So if they fit here, some fit and some neurotransmitters, as you can see, don't fit, right? And if it does fit, it's going to open up a gate. So these neurotransmitters are carrying that message from a sending neuron across the synapse to the receptor sites on the receiving neuron, and then that will open up the gates if the key fits. All right, so just to review everything, right? So first thing that happens, you have electrical impulse, right, called the action potential, travels from one neuron to another across a tiny gap known as the synapse. All right, so here's a sending neuron. You got action potential. This neuron is going to receive the information. Then here's a little blown up kind of what we just saw, right? So you got the vesicles, the neurotransmitters inside of it, the synapse or the synaptic gap in between, the axon terminal, the terminal buttons at the end, and then you have the receiving neuron, the dendrites are receiving these neurotransmitters, unlocking that neuron to fire. So when action potential reaches an axon terminal, it stimulates the release of neurotransmitter molecules from sacs called vesicles. These molecules cross the synaptic gap and bind to the receptor sites on the receiving neuron. The neurotransmitter molecules either excite or inhibit a new action potential in the receiving neuron. We'll talk about that in the next set of notes, because it can either excite it and make the neuron fire, or it can inhibit and prevent it from firing. So the other thing you should know is something called reuptake. All right, reuptake. So the sending neuron normally reabsorbs excess neurotransmitter molecules, a process called reuptake. So not all the neurotransmitters go to the next neuron, right? Some go back up and reattach to the neuron, just fires them. That is called reuptake. So like I mentioned earlier, neurotransmitters are like keys to a lock. All right, so these white things, these are going to be the neurotransmitter molecules. They each have a specific shape. Notice how if you walk around the house or like a school and you have different keys, they look the same, but actually they have a different shape inside. So receptor molecules have binding sites. So the neuron that's receiving the message has a binding site that the neurotransmitter is going to fit into like a key. So when neurotransmitter binds to the receptor, it opens up and positive ions rush in. All right, so let's see what that looks like. All right, so notice that you have the neurotransmitter fit the key perfectly. So see how this gate's closed? Well, then now this gate opened up, and you have all these positive ions rushing in, and that's going to excite and fire off the next neuron. So you need these, once again, these neurotransmitters fit into these keyholes, and if they fit perfectly or good enough, it's going to open them up, and then you have positive ions rushing in like you do here, and that's going to fire the neuron. So here's just a picture of different ones, how they can look differently. So a neuron has different receptor sites, and each one's going to fit a different key. Right? So depending on which neurotransmitter you have, this one's going to fit here, this one's going to fit here. So this one can't fit into this one because it's not the right key.